Today, we're going to be talking about Beam Therapeutics and how they utilize gene editing. Let's hop straight into it. Beam focuses on base editing. Well, to learn about base editing, we must first understand how the human genome works. Each DNA strand, or the genetic material that makes up who we are, has four bases: adenine, cytosine, guanine, and thymine. Now, these four bases pair with each other. Adenine always pairs with thymine, and guanine always pairs with cytosine. The average human genome contains over 3 billion base pairs, and even if one pair is misspelled, abnormal proteins will form. Abnormal proteins cause disease. Base editing can be used to solve this problem. There are a total of 5 different ways to base edit. First, we have gene correction, which directly repairs misspelled letters to restore the function of the gene. Next, we have gene modification, which inserts protective clinical variants to prevent the risk of disease. Thirdly, we have gene activation. This edits regulatory elements to reactivate gene expression. After that, we have gene silencing. In this form of base editing, stop codons, which are located inside of the messenger RNA, are edited to silence expressions. Lastly, we have multiplex editing, which edits multiple genome locations simultaneously. A protein that switches base letters are called base editors, or something that switches out the letters in the DNA bases. This protein is made of two components. The first component is a CRISPR protein, which is bonded to a guide RNA. The RNA leverages the CRISPR's protein's DNA targeting ability, but modifies it so the double strands of DNA do not break. The second component is a base editing enzyme called the deaminase, which chemically modifies the target DNA base. This combination allows the DNA base to be targeted and edited. Now, how does this base editor detect where a letter or base is misspelled? Well, it's actually quite simple. The CRISPR recognizes a complementary section inside the guide RNA. That's it. Now, let's get back to Beam. They believe that the modular and individual components of the base editors can be quickly rearranged to target specific diseases and create new therapeutic programs. The guide RNA portion of the CRISPR protein must be changed to retarget base editors to different genome locations. Then, by changing the deaminase, the exact base edited can be controlled. Now that we've talked a little bit about base editing, what are the advantages? Well, first, there is high efficiency editing without any need for template based homology directed repair. Two, there can be an avoidance of unwanted consequences of DNA strand breaks, such as frequent insertions and deletions. Lastly, you can create precise, predictable, and efficient genome outcomes at a targeted sequence. Beam Therapeutics hopes to cure many diseases through base editing and provide an easier life for countless people who suffer from these various diseases. Thank you. And please subscribe if you want to see more content videos like this one. Thank you for watching.